Okay, it's noon Central Time here in Houston, Texas. Welcome everybody to the restart of our monthly Center of Antimicrobial Resistance and Gulf Coast Consortium of Antimicrobial Resistance in the Texas Medical Center. I know we are in a very difficult time right now, and I want to take this opportunity to wish everybody the best. We want to be united in this front in our Houston infectious disease community, and I'm hoping that this will be a channel to do it. Just before we start, our next meeting will be in July. We'll be discussing our local efforts in the different institutions of COVID-19. So stay tuned, because that will be the time where we'll probably hit a higher peak in our community. For today, we're also making history. This is the first time that we do this. We have actually paired with this very strong Society of Young Scientists, the ASM Texas Medical Center chapter, has provided us with all the platforms and contacts to launch this effort together. As I mentioned to you, the power of collaboration in the Texas Medical Center is unparalleled, and this is a demonstration of this. We have a stellar speaker to start this series of online talks. Is Dr. Eleftherios Mylonakis, who is the Charles C.J. Carpenter, MD, Professor of Infectious Diseases, Professor of Medicine and Assistant Dean of Medicine at Brown University, and Professor of Molecular Microbiology and Immunology. Dr. Mylonakis is recognized for his research on the study of microbial pathogenesis and host responses. His studies have included clinical and laboratory approaches in the use of mammalian and invertebrate model hosts. The surrogate hosts fill an important niche in pathogenesis research and have provided us with a unique opportunity to identify novel antimicrobial compounds and study basic evolutionary conserved aspects of microbial virulence and host response. So Dr. Mylonakis is a very prestigious researcher, and he has found that all his research indicate that a common fundamental set of molecular mechanisms is employed by pathogens against a wildly divergent array of hosts. He's going to talk to us about the exciting research he's conducting on discovering antibiotics and new molecules. I also want to tell you that we are moving ahead for our January meeting, which is a 3-day meeting, to have this meeting online following these platforms. There will be a registration for that particular event, and we will sending information accordingly very soon. So I want to explain some of the technicalities in our partnership with the ASM TMC chapter. I want to hand it over to Dr. Aisha Khan, who is going to explain a little bit more of the questions and how you should interact. Aisha?
Hi, everyone. Thank you for being here. Again, this is an incredible opportunity, especially for early career scientists, to take on a more of a leadership role in ASM TMC and in our medical center microbiology community. So we are today broadcasting simultaneously on Facebook and Zoom. And if your colleagues are not here today and you want to make sure they see this, it's going to be uploaded onto YouTube. So we pasted the YouTube link to our channel, and that's where it'll be up. So keep in mind that you can ask questions at any point in the seminar as Dr. Mylonakis is talking, if you want to make sure especially that you don't forget by the end. And at the end, we will have a moderated Q&A where we'll select questions and ask, but you can post at any time in the Q&A box on the bottom of your screen. And also, one of our officers, Lynn, will post a link for MicroTalks, which is our monthly seminar series that anyone can give a talk at. And if you're interested in signing up, it's a different topic in infectious diseases every month, and the link is in the chat as well. And with that, thank you, and looking forward to hearing Dr. Mylonakis' work.
Hi, I hope that technology allows me to transmit to Houston from Providence, Rhode Island. Obviously, our thoughts are with you and with the healthcare workers who are fighting COVID-19. And needless to say, our thoughts and prayers are with every single healthcare provider and patient. I was asked to give you a break from COVID-19. I'll try to do just that and help you think about other infectious diseases. I offered to give a COVID-19 talk, but I was told that you would like this break. So we'll stay with mostly antibacterial compounds, and then we are going to maybe have some slides on COVID-19 afterwards. So the main vehicle of my presentation is going to be to introduce you to C. elegans as a model organism for host pathogen interactions and high throughput screens. So we'll start.
Dr. Mylonakis?
Yes.
I believe if you reshare your screen and not select optimization settings, it won't be blurred because it looks a little bit blurred right now. Rob, the administrator from Baylor, specified that in the chat for panelists.
Is this better?
Much better. Yep.
Much better, yes.
Okay. So those are the C. elegans worms, and they're about 1 millimeter in size, and that is going to be also the way to introduce you to some of the work that we have been doing. So the basic concept of this work has to do with the use of C. elegans as an in vivo screen model for the study and identification of new virulence factors. There are multiple reasons why you would like to do a screen using a whole animal model, as long as you can do it in high throughput. The main reason is that you can identify compounds and screen for toxicity at the same time that you are screening for efficacy. I'm not going to burden you, since this is an established model now that we have been using for about 20 years. I won't burden you with details of on the use of this model for high throughput screen. But I will proceed with some of the aspects of the technical screen that we are doing. So our goal is to do high throughput screening by imaging using an C. elegans worm. And at the bottom here, you're seeing how we can educate our microscopes to differentiate live versus dead worms. So essentially, if the worms die, we give them cytox. Cytox will only stay in the dead worms. So this allows us to visualize by microscopy and by screening by imaging the dead worms. Here you see dead worms receive placebo, and here you're seeing MRSA infected worms who are alive, and they were placed on vancomycin. So the way that the screen works is based on this approach where we use a publicly available screening by imaging algorithm in order to be able to evaluate the fluorescence that comes out of a well. That tells us how many dead worms are there versus the whole diameter of the well. So having told you that, then you can appreciate how this screening works. You're seeing here a 384 well plate. You're seeing here the controls, the negative control and the positive control. And you see everything that you see here is a worm that is dead. Only dead worms, as I said before, are stained with cytox. So essentially, if you have a well here where you have no fluorescence, i.e. you cannot see any dead worms, that means that all the worms are alive. So that means that the compound that you put inside that well has an antibacterial activity for somehow helped your worms survive MRSA infection. So essentially you get the worms, they are very small, 1 millimeter in size. You have a robot that only is going to dispense a exact number of worms, usually about 15 worms in each well. Then you're going to add your compounds, and then you're going to stain them after the infection process, then stain them, and then do screening by image. So I hope that I went through reasonably in a sensible way of understanding how we do our screens. And here are some of the results of our screen. Here is also a schematic of how the study works. So we have a compound library, then you have a toxic and non-toxic leads, and then we screen for them and you identify non-toxic leads, which we are going to take further. The main results of our screen were published in this Nature paper a couple of years ago. And that's the paper where I'm going to also use to identify the tools, and then from there go to more recent works that we have published. So at the time of that publication, we had screened a little more than 80,000 compounds, and we had identified 426 hits. Hits are those that we come through the screen. They are cherry-picked. That means we go back to them a second time around, they remain hits, and then we start them further. The first two compounds that I'm going to discuss are the two compounds listed here, the retinoids CD437 and CD1530. They are both retinoids. That means that they are both derivatives in some degree from vitamin A. So the basic molecular structure is that of vitamin A. And then the good thing about those retinoids, so why we like those compounds as primary compounds for our evaluation, was that we had two compounds that came through our screen. There was a third compound which was evaluated in the past and which it was publicly available, not as an antibacterial, but it was publicly available. And then there was a fourth similar retinoid which happened to have no activity. Although you see that all four of them, they have the similar structure. But you see a difference here, pretty obvious here. The hydroxymethoxy group that will come back later on. And three of them, they have activity against in the C. elegans model. And the fourth one, that is similar except of this tail here, has no activity whatsoever. So that is a good starting point. You have multiple hits and you have a negative control that looks the same. That is an ideal vehicle in order to start exploring how far you can go with those compounds. So the first thing that, that you do is then you confirm your findings from the screen. Screens are exciting, are good, but they're not publishable information. You would like to take the information. Usually my experience with screens, and I have done quite a few of those in different situations, the best that you can get is that you get a 30% noise. So you should not get too excited when you have a hit. Then you want to cherry pick and do a very careful screen where you have a dose-dependent evaluation of the survival of the worms. So that allows you to go through different increasing concentration and see how much the worms survive during the infection. That gives you a better confirmation of the hit results. And then you do in vitro screen to see if the compound indeed has some direct antibacterial activity, and you see a time, this is a straightforward microbiology time kill assay, and you see here the killing that happens pretty quickly with the 3 of the 4 compounds that I mentioned before. Now, the good thing about working in a hospital, and that's is that we have plenty of isolates, so we can always go back and identify and screen for efficacy. And we found that those compounds only had activity against the gram-positives. They had some activity against Enterococcus staph laboratory strains, but now no activity against the gram-negatives. Then we took a large selection and a collection of clinical isolates, a few dozens. Actually, we have almost 1,000 now. And we confirmed that the finding, the MIC that we see is not unique to the laboratory strain MW2, but we see the same screen, the same finding with all different clinical compounds and clinical isolates. That is exciting, but this is just the beginning of trying to identify what the compounds really do against the bacteria. The exciting part of antimicrobial drug discovery comes from trying to identify the target. This is particularly important because even if the compound is very toxic or turns out not to be druggable, then identifying a compound will allow you to always screen against that, that compound and then against that target and find other compounds that might be better and more druggable. And the easiest way of trying to identify the mode of action is the following. You take the bacteria that you are studying, you expose it in progressively increasing concentrations of your antibiotic, and then you try to breed resistance. Here is what we did with the two retinoids, and the control was ciprofloxacin. And you see here that ciprofloxacin very quickly, the MRSA strain that we used, developed resistance within 100 days. Unfortunately, or fortunately, we found that the Staphylococcus was really unable to develop resistance to the two retinoids. And that was both very exciting because it showed that its resistance, development of resistance is very rare, but also it was made our life a little miserable because there was, we couldn't identify the target very easily. Nevertheless, we took those peaks here, the progressive, this MIC that went from 1 to 2, from 2 to 4, that is the maximum MIC that we were able to find. We took those peaks and we did a whole genome sequencing, and we found that those mutations that really increased the MIC even incrementally, all of those mutations happen to be on the genes that are associated with the membrane. That was particularly interesting. We didn't know what to make out of this because working with the membrane is always a challenge. And this confirms what I told you before, but we wanted to make sure that that was the case. We took mutants that others had published with mutations on those genes, and indeed we found that if you, the strains that had the mutation on those genes that are associated with membrane had a little higher MIC. As I said before, this is exciting, but the membrane, working with the membrane, is really challenging. You cannot really do very easily molecular genetics, and difficult to find a lot of literature on the topic. The first thing that we did afterwards was to confirm indeed that that was the target of our compounds. The confirmation is pretty straightforward. It's a fluorescence assay where the more permeable the membrane is, the more fluorescence goes in, and you just measure the fluorescence. And you see here that those two retinoids really increase the fluorescence really quickly of MRSA strains. And then another confirmation is by electron microscopy. You see here those abnormalities on the membrane. So that gives us a lot of confidence that really those retinoids target the microbial membrane. Okay, so the next challenge is to try and identify where exactly on the membrane they act, or try to find better the interaction between the membrane and your compound, in this case, the retinoids. Thankfully, there is one way that you can do microscopy and study how a compound interacts with a membrane. That is called creating GUVs. GUVs are large membrane-like giant vehicles that are big enough for microscopy. So essentially how it's done, you take the components, which is DOPC and DOPG, 7:3 ratio, and you use established protocols of electrophoresis in order to try and make out of that lipid giant vesicles that look like a membrane, and they are many times larger than a microbe. The good thing about those is that you can do microscopy on those, and those are big enough, and you can see here that once you expose, you have those GUVs, you expose them to different components, different compounds, and you see here that you start to see on those that are exposed to the retinoids. I hope that you can appreciate the small clumps that they develop. Those are just vehicles with the membrane there. There is nothing else to interfere. So that shows that the compounds, in this case, the two retinoids, are able to develop and make those small collections. That is additional proof that the compounds act on the membrane. And this is not the first compound that, or the first compounds, of course, that act on the membrane. Of course, daptomycin is a compound that has been associated with making those same clumps on the membrane. Although the molecular structure of daptomycin, of course, is vastly different from our compounds. Here is a control. Others have done it also, showing those clumps made by daptomycin and how it's done there. Now, if you want to get in more detail of how the chemical interacts, the compound interacts with the lipid bilayer, there is a computer system where it isolates one molecule and tries to see how that molecule interacts with the membrane. Here you see how the CD437, CD1530, the two positive retinoids that came through our screen, how they can interact. They use that hydroxy moiety, part of the moiety that I told you before, that is the difference from adaptalene, and they anchor to the lipid bilayer, and they use it in order to push through, while the adapalene, which has a methoxy group there, cannot really anchor and cannot penetrate. This is another way of showing that penetration and how quickly it happens. As you have seen before, it doesn't happen at the same time. Interestingly, the membrane is a very intriguing factor to develop molecules against and develop a new antibiotics against, because it's unique in bacteria, but also the problem here is that a lot of the compounds that act on the bacterial membranes act also on the mammalian membranes. So we took our compounds and we screened for toxicity against a series of mammalian cells, starting from red blood cells, going to a number of different cell lines. And essentially we found that the interaction of retinoids with membranes, it was limited to the bacterial membrane. You see here that, for example, I have here the hemolytic activity, that there is almost no hemolytic activity on the red blood cells. And also we have published a couple of protocols on studying persisters in a more high throughput screen. And we use those protocols in order to identify the activity against MRSA persisters. Why did we go to MRSA persisters? Well, persister cells are dormant cells. and they are associated with the relapse that we see in staphylococcal infections. Staph aureus has a unique capability, more than most other bacteria, in order to develop, to take that dormant state that is described as persister cells. And that's probably why when we are on clinical service, we need to treat patients with staphylococcal infections much longer in order to get rid of the infection compared to patients who have E. coli or some other infection where there is less likelihood of developing these persister cells. Essentially, up to 90% or 100% of the cells of Staph aureus can become persisters, dynamically can become persisters, while for E. coli or most other bacteria, that is 10% or less. A huge capability of taking the dormant cell. Now, if you have a compound that works against the membrane, then this is a compound that probably works against persisters because the membrane is there. There is no way that the bacteria can find a way to hide the membrane. Even if they become dormant, the membrane will stay there. So indeed, we took those protocols that I mentioned before, and we evaluated the hypothesis that those compounds have activity against persister cells. So we made persisters. That is very easy protocol to do. You essentially blast the bacteria with high concentrations of an antibiotic, and then you expose them to the compound, those that are survive. And you see here that those two retinoids that I have told you for the last half an hour or so, they really have an activity and they are able to decrease the survivability of CFUs per ml of the bacteria. Well, the negative control, Adaro 10, which, as I told you before, and as I showed you, is unable to penetrate, cannot really, of course, kill the persistent cells. Here is the same depiction using different strains of different clinical isolates. And again, you see here that if you take those persistent cells, you make persistent cells and you expose them to your retinoids in this case, then you have essentially are able to destroy the persistent cells. We found also that as others have described before, compounds that work on the membrane and work against persisters sometimes synergize with gentamicin. This is the a straightforward protocol about how to do synergy. I won't go into this. Bactericidal activity when combined with gentamicin. Then you have to go and try to optimize your antibiotics, your hit compounds. Try to increase efficacy, decrease toxicity. We didn't have a lot of toxicity. We had pretty good efficacy with our compounds. Nevertheless, usually pharmaceutical companies, when they go from a hit to evaluate further and try to make analogs, they usually aim for making anywhere from 500 to 1,000 or more chemical compounds that would help also identifying the mode of action. And here, of course, as an academic lab, we cannot really ask for our medicinal chemist collaborators to do hundreds of compounds. Instead, they were able to take the molecular dynamics that I showed you before, the understanding of the molecular dynamics and how which part of the molecule, as you see here, is important for the anchoring and the penetration, and try to work from there and try to make analogs. And indeed, they made a handful, about 17 analogs, and 2 of them, analog 2 and analog 19, were able to have even better activity as far as MIC and rapid killing of persisters compared to the parent retinoid. Here is repeating the same assays and the same evaluation that we did before. The analog 2, which is depicted here, was able to anchor and penetrate in a way that was faster in the beginning than the parent strain of CD437. I won't let the whole video play, but I will take you to the punchline, which is then you would like to try and study those findings in an animal model. And there is a very good thigh mouse model that allows the evaluation of persister cells. Essentially, it's a thigh model where you inject the MRSA into the thighs of mice, and then you evaluate your compounds afterwards. The others who developed this model have shown that almost 100% of the MRSA or Staphylococcus aureus bacteria here in the thigh of the mouse become persisters. And indeed, we found that the compounds, analog 2 in this case, especially when it was combined with gentamicin, was able to decrease the CFUs of cells inside the mouse of thigh, inside the thigh of mice. Also, we checked for toxicity and had the combination of gentamicin with the analog 2 had no effect in liver function tests or the renal function. About the same time, we did similar studies with another compound called anti-DGPA. I'm not going to go into detail, but I'm going to just build from what I told you before, because you know the tools, as I said before, the tools of what we do in order to evaluate our compounds. I tried to use the retinoids in order to describe them in detail, but you can understand here that what we did, it's the same electron microscopy. We found that it affects the membrane, the same GUVs here, the same molecular dynamics here. What it adds here to our understanding is that the mechanism of action allowed us to have different chemical structures of anti-DGPA. Why was that important? That was important because in a future study we found bithionol, and the bithionol is an antiparasitic agent that we found it to have efficacy again against the membrane. We were very interested in this, and there are two important developments that we made in this work. The first one was, of course, describing bithionol, but the second important step was that we found that bithionol penetrates the membrane, as I showed you before, with the retinoids, causes the GUV to lyse. And also, we associated the membrane activity with fluidity. So what we found is that there are a number of compounds, and we used for that the anti-DGPA analogs and the bithionol analogs. Those two groups of analogs, they had a variety of activity. And we were trying to figure out why there was such a difference in activity. All of the analogs, the chemical analogs, they were able to increase the permeability. You remember that I showed you that the fluorescence goes inside the cell, you measure the fluorescence, and this is the first sign that very easy fluorescent assay that will tell you that your compound acts on the membrane. However, not all of them, although all of them increase the permeability, not all of them had a killing of antibiotic tolerant MRSA, the persister cells that I told you before. Why? Well, the missing link we found between the permeability and the killing was fluidity. So the structure, it's not enough to allow penetration of antimicrobials, but also it has to have a change in the architecture of the lipids of the membrane. And how do you figure it out? You figure it out by evaluating, there is a fluorescence assay again that evaluates fluidity by the change of the water molecules inside the lipids, and that's what it essentially measures. Lordan is the name of the chemical structure made by an Argentinian chemist many years ago. And in any case, you see the fluorescence, which is a depiction of fluidity, and the concentration that you need in order to kill dormant cells. And you see here that every time that you have ability to kill dormant cells, you have also a decrease in fluidity. And putting those things together, this is a paper that is now in press, and that discusses another compound that we have developed. It's called PQ401. You see the chemical structure of PQ401, and PQ041 is unique because it's neither cationic nor amphipathic. So it doesn't have the chemical difference, the electrophysiological difference that will allow it to penetrate and associate with membrane. Instead, it has to rely on its ability to anchor through the chemical structure. So, it can penetrate better when it's in a neutral form. I think this is the slide, yes. You see here that the neutral form actually penetrates much better than the proteinated or deprotonated forms of the analogs of this molecule. Why is this important? This is important because the more neutral the compound is at 7.4 pH, the more likely it is that it's going to be well tolerated and not very toxic. So what I try to take you through here is to show you that the C. elegans MRSA infection assay, it can provide a unique way to evaluate compounds and find antimicrobials and also anti-infectives that may not have a direct antimicrobial activity, but have an activity during in vivo screens. We propose this as another way of drug discovery that is different from the traditional screens. You see here the high throughput in vitro screen that we usually use as a traditional first step, where we take a Acinetobacter or MRSA or something else, and we go and we evaluate the efficacy of thousands of compounds. Usually, most pharmaceutical companies have about 100,000 compounds that they evaluate, but that gets you a lot of hits that are very toxic. So you have to rely on an endless series of optimization steps that here, when you have a host inside the well, those evaluations for toxicity and potency and in vivo efficacy can happen at the very first step of the screen, and then help you evaluate even anti-membrane compounds that have low toxicity. So quickly, I'm going to acknowledge the people that did most of the work here. It's my lab and Woo-Shin Kim, who is now assistant professor in Korea, who was the person that was the first author of the retinoid paper, and my group. Here is it's a very powerful story of one of our former trainees who became public. Tori Kinamon, her first admission was with MRSA infection. She was a gymnast at Brown, first year, and she was admitted, completely healthy, was admitted to the ICU for 4 weeks because of MRSA infection and sepsis. And she recovered from that after 4 operations, and she was able to come to the lab and help our discovery. However, I think that at this point, we are all stressed as healthcare workers looking at the COVID-19 situation. So I promise that I'm going to give 5 minutes at the end with our thoughts and some lessons that we learned from COVID-19. The first lesson that we learned here, we have a very small proportion of Hispanic or Latinos, and the proportion of that among them who came to the hospital was twice that of the general population. So one of the first things that we did is that we sent our ID docs who are fluent in Spanish to talk to the radios and to the TV stations and try to educate that population. That was very important to us. We didn't figure it out until a month into the process how common it was for non-English speaking population to not be as well informed about it. The second association that was very important for us, and we changed our approach to prioritize patients, was associated with obesity. For us, looking at our first wave of patients, obesity with BMI over 35 was the single most important risk factor for ICU admission. And we changed our protocols to include either BMI over 35 or BMI over 30 plus heart disease. In our experience, remdesivir really made a difference in mortality. Although that was not statistically significant, there was a strong trend, and it was very safe as far as AKI. So that was a result of about 120 patients on remdesivir from the clinical trials that we did here. This is a single center analysis, and this is something very important. Houston is the forefront of I think, plasma. That is very exciting. One thing that we learned and helped us is the engagement of the Antimicrobial Stewardship group. The Antimicrobial Stewardship group was important in evaluating, in those days, the hydroxychloroquine. They had a protocol. Of course, hydroxychloroquine didn't make a difference in the survival or any other parameter, but engaging antimicrobial stewardship in those endeavors really made a strong difference in our ability to evaluate new protocols. So learning how to evaluate, how to optimally use antimicrobial stewardship was something else that we learned during the process. What was very helpful to us also is that we needed to establish a post-COVID-19 discharge clinic where everybody who was discharged from the hospital with COVID-19 received a call and an evaluation through a standard questionnaire within 48 hours from discharge. That helped a lot with readmissions and gave a lot of confidence to the hospitalists to discharge patients, because they knew, and the emergency room, because they knew that the ID docs will follow up by televisit within 48 hours. We found it to be very helpful. Also, looking ahead, we looked at the hotspots of looking to the influenza season. The take-home message here is that the influenza and COVID-19 happen in the same areas. Here is the Providence, which is the larger city in Rhode Island. And we found that the hotspots, which we evaluated from influenza, that are very small neighbor level hotspots, about less than about half a mile, 900 meters. They happen in the same place where, for influenza and COVID-19. So in influenza, there are three groups of epidemics. There are the children, the adults, and the elderly. In the COVID-19, there are the same hotspots, but there are two epidemics, different epidemics, not associated with one with another, mostly, but the hotspots are the same. So bottom line, we need to vaccinate people for influenza. And if you have a free day or some time, a very powerful book is The Plague by Camus that was published in 1947. And the only means of fighting a plague, they say, it's common decency. And what do you mean by common decency? It means to do your job. So healthcare workers doing our job during this epidemic can help us fight the plague that has come upon us. There are a lot of heroes of healthcare workers. I think it's sometimes it feels trivial to talk about membrane and activity in publications. in the face of heroes who gave their lives in order to help others in the form of the pandemic. And all of us as healthcare workers are in awe for some of our colleagues and their abilities to come up and respond to this challenge. I would like to thank you all for the good things that you're doing, helping your patients, and grateful for the opportunity to talk to you today.
Thank you very much, Eleftherios. It's fantastic. Now, Aisha is going to moderate the Q&A section.
Thank you, Dr. Mylonakis. That was really great. I'm going to start with the first question we got, which was from Cesar, actually. Why do you think, or do you have an idea, and have you looked into it, if aderatine, why does it not have activity?
Yes, it's unable to anchor because of the methoxic versus hydroxy group. Just one molecule, just one change makes all the difference in the activity.
Okay, and I guess in a follow-up to that, actually, because a lot of what you spoke about was interaction between the molecule and the membrane and affinity of charges, it can be pretty hard to discriminate between what the mechanism of death is and if membrane fluidity changes and membrane permeabilization is like an aftermath of cell death. So like with daptomycin, for example. So have you, what are your thoughts on that? And have you explored how to distinguish between what is the actual precise mechanism of killing as opposed to the general membrane permeabilization?
Yeah, well, the understanding that we have right now is that they anchor on the lipid component of the membrane, and they penetrate and they change the architecture. So that's as far the architecture of the membrane, causing change in fluidity, permeabilization, and then influx of water and electrolytes that kill the cell. So that's as far as we have gone so far, but still the more precise mechanism of action is yet to be discovered, and I'm sure that it's not going to be one for all the compounds, but different interaction for them.
And on that note about mechanism of action, have you looked to see where some of these molecules bind in the cell?
Yes, from the molecular dynamics, we believe that they bind to the lipid part of the membrane and the outer part.
But is there specific localization, like the division septum, for example, with daptomycin?
No, we haven't looked at this.
The next question was, they're curious about your screen, your compound screen, that and the question was, do you find compounds that improve worm survival but don't have any effect inhibiting bacterial growth when they're just used alone?
Can you repeat compounds that work in the worm, but they don't work in vitro?
Correct, yeah. Compounds that improve worm survival, but don't actually inhibit bacterial growth.
Yeah.
And yeah, anything to
Well,
do with
those we do have those, and those are the most difficult compounds to study because those might interact or might influence the immune response of the worm or might decrease the virulence of the bacteria. So those are the most exciting compounds because those compounds you cannot find from an in vitro screen, but those compounds we don't have we are developing the tools to study them, and we have ways to go there.
That was the follow-up that also Fan asked, that if these compounds that are boosting the host animal immunity, like, do you think that with these compounds, just given the similar mechanisms of action with other cell membrane disrupting agents, that they could be used as adjuvants potentially to existing antibiotics?
Yeah, well, the compounds that work through modulating immunity would be adjuvants, but difficult to make that claim when we have a total of 0 publications of studies coming out of that. We are hoping to have something at some point in a year or so.
Okay, next question was a more general question, I think, that a lot of us are sometimes interested in if we're not involved in clinical trials, which is, are any of your compounds on their way to clinical trials, and how long does it actually take for a discovery of a promising compound like this to make it to the bedside?
They say 5 to 10 years at least, but honestly, this is quite different from what it used to be. It used to be that pharmaceutical companies would take those compounds from discovery through clinical trials and then just go through the process. Here, the whole process is fragmented. Pharmaceutical companies don't do any screening anymore. Some biotechs will pick up, if you have good mouse data, will pick up your studies. So you are in an academic lab trying to develop collaborators and move things along and get funding from NIH and try to advance it. So how that 5 to 10 years is going to translate in this environment of COVID-19, of limited resources, of high deficits, decreased probably NIH budgets, it's something that we'll all suffer through.
And the next one was in the Q&A as well from Jalen Hurdle. These types of membrane active compounds often are hydrophobic. So how does that affect their pharmacokinetics? And especially, is it a liability and something to consider when we're treating systemic infections?
Yes, they have a tendency to bind with proteins. We haven't done PK/PD assays. This is actually what we're doing right now in order to help them develop. And that is one of the reasons why we're trying to make enough analogs that we address that, chemical analogs.
What about thinking about the mechanism of resistance in this case, when we're looking at diverse molecules where, you know, the targets in terms of the precise lipids might change, and have you looked into what that mechanism of resistance might be, or what the evolutionary trajectory to resistance could be?
So far, we haven't, fortunately or unfortunately, as I said, we haven't been able to breed the resistance. I'm sure that bacteria can develop resistance to everything, but so far we haven't been able to get that resistant isolate to evaluate it further.
The next one was a follow-up on the connection between these compounds and daptomycin. So daptomycin, now the mechanism of action has evolved from before it was pore forming model, and now it's more about how it can mislocalize proteins that are involved in essential cell processes like cell wall synthesis, cell division, et cetera. So in your case, did you look at peptidoglycan mechanism and if the peptidoglycan structure or metabolism is affected at all?
We haven't looked at that. I didn't present daptomycin as having the same mechanism of action. I present in the context of showing that it has the same effect in GUVs. So that was the context that I presented. But that is a very good point. I would like to thank the person who made that suggestion.
And the next question, for the synthetic compound, when you said that the chemical composition of that compound was unique from all of the other identified hits, can you give, and the fact that it has a unique chemical interaction with the lipid bilayer. So can you give a little bit more insight on what you meant by the chemical composition being unique and the chemical interaction itself being unique?
Yeah, that was a point that I made really quickly, and thank you for letting me go back to it. So what I was trying to make the point is that it's different, unique. Unique is a big word. It's different from the other chemical structures, and also this is a compound that has a unique feature from the others. That at 7.4 pH, it's usually neutral. And if you go cationic or anionic forms, then you lose activity as far as the interaction with the membrane. So that allows you to have a potential hit that will be in a neutral electric environment, a charge able to associate and work on the membrane. That's, I hope that that gives you enough context to clarify what I meant.
Yeah, and Cesar, I see him nodding because I think even with all of the work that we do with antimicrobial peptides, the charge is the biggest unique interaction that we focus on, especially when it comes to a mechanism of action.
That's exactly right. That's our concern with peptides and our difficulty with peptides.
And a follow-up on the toxicity issues that you talked about. Why was there no effect on mammalian cells?
Yeah, well, as far as I can tell you is that what we know is that if you change the way that we challenge that, we evaluate that, it was using, again, the molecular dynamics, now having the chemicals versus the mammalian membranes that has a lot of cholesterol, about 30% of cholesterol. And we found that the compounds that are non-toxic are unable to associate and penetrate the mammalian membrane with the cholesterol. So that is the main difference that exists in the membrane, and that's the main effect that does not cause any toxicity there. It's a different membrane, compounds that shown to be non-toxic, they are unable to penetrate the mammalian membrane while they have some selectivity against the bacterial one.
Okay, and I think that answers all of the questions that we got. If anyone has any final questions, you can post it in the chat, and if not, then Dr. Arias will close us out today.
So thank you, Eleftherios. Fantastic talk. You know, it's a topic that we are very much into it as well. So we were very looking forward to have you here in Houston. So I'm sorry, but I promise that, you know, when things calm down, you know, I owe you an invitation to Houston. For everybody, I want to announce that our next talk is going to be on July 20th. We are hoping to have a 1.5 hour discussion. Is our normal stewardship session, and this is going to be focused on the different experiences of our hospitals over COVID-19, and it's going to be moderated by Dr. Ed Septimus and Kevin Garrett, who is putting a really representatives from all hospitals. As I mentioned to you before, even from this morning in Twitter, I think this is the time to get together to understand what other people are doing, our neighbors are doing, and I think that will help all of us to treat our patients and in the most critical moments that we're living too. I want to thank Aisha and all the ASM TMC team for this wonderful partnership that we hope to continue. You know, doing this online is been a great success. All the participants and the questions, thank you very much. Again, thank you, Eleftherios, and I'll see you next time. Thank you very much.
Thank you.